Class 12, Chapter 11, Biotechnology Principles and Processes. Biotechnology deals with techniques of using live organisms or enzymes from organisms to produce products and processes useful to humans. In this sense, making curd, bread, or wine, which are all micro-mediated processes, could also be thought as a form of biotechnology. However, it is used in a restricted sense today to refer to such of those processes which are genetically modified organisms to achieve the same on a large scale. Further, many other processes or techniques are also included under biotechnology, for example, in vitro fertilization leading to a test tube baby, synthesizing a gene and using it, developing a DNA vaccine, or correcting a defective gene are all part of biotechnology. The European Federation of Biotechnology (EFB) has given a definition of biotechnology that encompasses both traditional view and modern molecular biotechnology. The definition given by EFB is as follows: the integration of natural science in organisms, cells, part thereof, and molecular analogues for products and services. 11.1 Principles of biotechnology. Among many, the two core techniques that enable birth of modern biotechnology are first genetic engineering techniques to alter the chemistry of genetic material DNA and RNA to introduce these into host organism and thus change the phenotype of the host organism. Second, maintenance of sterile microbial contamination free ambience in chemical engineering processes to enable growth of only the desired microbe or eukaryotic cell in large quantities for the manufacture of biotechnological products like antibiotics, vaccine, enzymes, etc. Let us now understand the conceptual development of the principles of genetic engineering. You probably appreciate the advantage of sexual reproduction over asexual reproduction. The former provides opportunities for variation and formulation of unique combinations of genetic setup, some of which may be beneficial to the organism as well as the population. Asexual reproduction preserves the genetic information while sexual reproduction permits variation. Traditional hybridization procedures used in plants and animals breeding very often lead to inclusion and multiplication of desirable genes along with the desired genes. The techniques of genetic engineering, which include creation of recombinant DNA, use of gene cloning, and gene transfer, overcome this limitation and follow us to isolate and introduce only one or a set of desirable gene without introducing undesirable gene into the target organism. Do you know the likely fate of the piece of DNA which is somehow transferred into an alien organism? Most likely, this piece of DNA would not be able to multiply itself in the progeny cells of the organisms, but when it gets integrated into the genome of the recipient, it may multiply and be inherited along with the host DNA. This is because the alien piece of DNA has become part of the chromosome, which has the ability to replicate. In the chromosome, there is a specific DNA sequence called the origin of replication, which is responsible for initiating replication. Therefore, for multiplication of any piece of DNA in an organism, if it needs to be a part of chromosome which has a specific sequence known as origin of replication. Thus, an alien DNA is linked with the origin of replication so that the alien piece of DNA can replicate and multiply itself in the host organism. This can also be called as cloning or making multiple identical copies of any template DNA. Let us now focus on the first instance of the construction of an artificial recombinant DNA molecule. The construction of the first recombinant DNA emerged from the possibility of linking a gene encoding antibiotic resistance with a native plasmid autonomously replicating circular extrachromosomal DNA of Salmonella typhimurium. Stanley Cohen and Herbert Boyer accomplished this in 1972 by isolating the antibiotics resistant gene by cutting out a piece of DNA from a plasmid, which was responsible for conferring antibiotic resistance. The cutting of DNA at specific location became possible with the discovery of so-called molecular scissors restriction enzyme. The cut piece of DNA was then linked with the plasmid DNA. These plasmid DNA act as vector to transfer the piece of DNA attached to it. You probably know that the mosquito acts as an insect vector to transfer the malarial parasite into human body. In the same way, a plasmid can also be used as vector to deliver an alien piece of DNA into the host organism. The linking of antibiotic resistant gene with the plasmid vector become possible with the enzyme DNA ligase, which act on cut DNA molecule and joins their ends. This makes a new combination of circular autonomously replicating DNA created in vitro and is known as recombinant DNA. When this DNA is transferred into Escherichia coli, a bacterium closely related to Salmonella, it could replicate using the new host DNA polymerase enzyme and make multiple copies. The ability to multiply copies of antibiotic resistant gene in E. coli are called as cloning of antibiotic resistant gene in E. coli. You can hence infer that there are three basic steps in genetically modifying an organism: identifying of DNA with desirable gene first, second, introduction of the identified DNA into the host, third, maintenance of the introduced DNA in the host, and transfer of the DNA into its progeny. 11.2 Tools of recombinant DNA technology. Now we know that from the foregoing discussion that the genetic engineering or recombinant DNA technology can be accomplished only if we have the key tools: restriction enzymes, polymerase enzyme, ligase, vector, and the host organism. Let us try to understand some of these in details. 11.2.1 Restriction enzyme. In the year 1963, the two enzymes responsible for restricting the growth of the bacteriophage in Escherichia coli were isolated. One of these added methyl groups to DNA while the other cut DNA. The latter was called restriction endonuclease. The first restriction endonuclease, hint second, whose functioning depending on a specific DNA nucleotide sequence was isolated and characterized 5 years later. It was found that hint second always cut DNA molecules at a particular point by recognizing a specific sequence of 6 base pairs. The specific base sequence is known as the recognition sequence for hint second. Beside hint second, today we know as more 900 restriction enzymes that have been isolated from over 230 strains of bacteria, each of which recognize different recognition sequence. The convention for naming these enzymes is the first letter of the names comes from the genus and the second letter comes from the species of the prokaryotic cell from which they were isolated. Example: EcoRI comes from Escherichia coli RY13. In EcoRI, the letter R is derived from the name of strain. Roman number following the names indicated the order in which the enzymes were isolated from the strain of bacteria. Restriction enzymes belong to a larger class of enzymes called nuclease. These are of two kinds: exonucleases and endonucleases. And exonucleases removes nucleotide from the ends of the DNA, whereas endonucleases may cut at specific positions within the DNA. Each restriction endonuclease functions by inspecting the length of a DNA sequence. Once it finds its specific recognition sequence, it will bind to the DNA and cut each of the two strands of the double helix at specific point in their sugar phosphate backbone. Figure 11.1. Each restriction endonuclease recognizes a specific palindromic nucleotide sequence in the DNA. Do you know what palindromes are? These are groups of letters that form the same word when read both forward and backward. Example: Malayalam, as against a word palindrome where the same word is read in both directions. The palindrome in DNA is sequence of base pairs that read same on the two strands when orientation of reading is kept the same. For example, the following sequences reads the same on the two strands in 5' to 3' direction. This is also true if read in the 3' to 5' direction. Restriction enzyme cut the strand of DNA a little away from the center of the palindrome sites, but between the same two bases on the opposite strand. This leaves single-stranded position at the end. There are overhanging stretches called sticky ends on each strand. Figure 11.1. These are named so because they form hydrogen bonds with their complementary cut counterparts. The stickiness of the ends facilitate the action of the enzyme DNA ligase. Restriction endonucleases are used in genetic engineering to form recombinant molecules of DNA which are composed of DNA from different source or genome. When cut by the same restriction enzyme, the resultant DNA fragments have the same kind of sticky ends, and these can be joined together end to end using DNA ligase. Figure 11.2. Figure 11.2 diagrammatic representation of recombinant DNA technology. You may have realized that normally, unless one cuts the vector and the source DNA with the same restriction enzyme, the recombinant DNA molecule cannot be created. Separation and isolation of DNA fragments. The cutting of DNA by restriction endonuclease result in the fragments of DNA. These fragments can be separated by a technique known as gel electrophoresis. Since DNA fragments are negatively charged molecules, they can be separated by forcing them to move towards the anode under an electric field through a medium or matrix. Nowadays, the most commonly used matrix is agarose, which is a natural polymer extracted from a seaweed. The DNA fragments separate resolve according to their size through sieving effect provided by a agarose gel. Hence, the smaller the fragment size, the further it moves. Look at the figure 11.3 and guess at which end of the gel the sample was loaded. Figure 11.3, a typical agarose gel electrophoresis showing migration of undigested LAN1 and digested set of DNA fragments LAN2 to 4. The separated DNA fragments can be visualized only after staining the DNA with a compound known as ethidium bromide followed by exposure to UV radiation. You cannot see pure fragments in a visible light, and without staining you can see bright orange color band of DNA in the ethidium bromide stained gel exposed to UV light. Figure 11.3, the separated bands of DNA are cut out from the agarose gel and extracted from the gel piece. The steps known as elution. This and the DNA fragments purified in this way are used in construction recombinant DNA by joining them with cloning vector. 11.2.2 Cloning vectors. You know that plasmid and bacteriophage have the ability to replicate within bacterial cells independent of the control of chromosomal DNA. Bacteriophage, because of their high number of cells, have very high copy numbers of their genome within the bacterial cells. Some plasmids may only have 1 or 2 copies per cell, whereas others may have 15 to 100 copies per cell. Their number can go even higher. If we are able to link an alien piece of DNA with bacteriophage of plasmid DNA, we can multiply its number equal to the copy number of the plasmid or bacteriophage. Vectors used at present are engineered in such a way that they help easy linking of foreign DNA and selection of recombinant from non-recombinants. The following are the features that are required to facilitate cloning into a vector. First, origin of replication, ORI. This is a sequence from where replication starts, and any piece of DNA when linked to the sequence can be made to replicate within the host cell. This sequence is also responsible for controlling the copy number of linked DNA. So if one wants to recover many copies of the target DNA, it should be cloned in a vector whose origin support high copy number. Second, selectable marker. In addition to ORI, the vector requires a selectable marker which helps in identifying and eliminating non-transformant and selectively permitting the growth of the transformants. The transformation is the procedure through which a piece of DNA is introduced in a host bacterium. You will study the process in subsequent section. Normally, the genes encoding resistant to antibiotics such as ampicillin, chloramphenicol, tetracycline, or kanamycin, etc. are considered useful selectable markers for E. coli. The normal E. coli cells do not carry resistance against any of these antibiotics. Third, cloning sites. In order to link the alien DNA, the vector needs to have very few, preferably single recognition sites for the commonly used restriction enzyme. Presence of more than one recognition site within the vector will generate several fragments which will complicate the gene cloning. Figure 11.4. The ligation of alien DNA is carried out a restriction site present in one of the two antibiotic resistance gene. For example, you can ligate a foreign DNA at a BAM-H1 site for tetracycline resistant gene in a vector PBR322. The recombinant plasmid will lose tetracycline resistant due to insertion of foreign DNA but can still be selected out from non-recombinant ones by plating the transformants on ampicillin containing medium. The transformant growing on ampicillin containing medium are then transferred on a medium containing tetracycline. The recombinant will grow in ampicillin containing medium but not on that containing tetracycline, but non-recombinant will grow on the medium containing both the antibiotics. In this case, one antibiotic resistant gene helps in selecting the transformant, whereas the other antibiotic resistant gene gets inactivated due to insertion of alien DNA and helps in selection of recombinant. Selection of recombinant due to inactivation of antibiotics is a cumbersome procedure because it requires simultaneously plating on two plates having different antibiotics. Therefore, alternative selectable markers have been developed which differentiate recombinants from non-recombinants on the basis of their ability to produce colors in the presence of the chromogenic substrate. In this, a recombinant DNA is inserted within the coding sequence of an enzyme beta-galactosidase. This results into inactivation of the enzyme, which is referred to as insertional inactivation. The presence of chromogenic substrates give blue-colored colonies. If the plasmid in the bacteria does not have an insert, presence of insert result into insertional activation of the beta-galactosidase and the colonies do not produce any color. These are identified recombinant colonies. Fourth, vectors for cloning genes in plants and animals. You may be surprised to know that we have learned the lessons of transferring genes into plants and animals from bacteria and virus, which have known this for ages. How to deliver genes to transform eukaryotic cells and force them to do what the bacteria or virus want. For example, Agrobacterium tumefaciens, a pathogen of several dicot plants, is able to deliver a piece of DNA known as tDNA to transform normal plant cells into a tumor and direct these tumor cells to produce the chemicals required by the pathogen. Similarly, retrovirus in animals have the ability to transform normal cells into cancerous cells. A better understanding of the art of delivering genes by pathogens in their eukaryotic host has generated knowledge to transform these tools of pathogen into useful vectors for delivering genes of interest to humans. The tumor-inducing TI plasmid of Agrobacterium tumefaciens has now been modified into a cloning vector which is no more pathogenic to the plant but is still able to use the mechanism to deliver genes of our interest into a variety of plants. Similarly, retrovirus have also been disarmed and now used to deliver desirable genes into animal cells. So once a gene or a DNA fragment has been ligated into a suitable vector, it is transferred into a bacterial, plant or animal host where it multiplies. 11.2.3 competent host for transformation with recombinant DNA. Since DNA is a hydrophilic molecule, it cannot pass through cell membrane. Why? In order to force bacteria to take up the plasmid, the bacterial cells must first be made competent to take up DNA. This is done by treating them with a specific concentration of a divalent cation such as calcium, which increase the efficiency with which DNA enters the bacterium through pores in its cell wall. Recombinant DNA can then be forced into such cells by incubating the cells with recombinant DNA on ice, followed by placing them briefly at 42 degrees Celsius heat shock and then putting them back on ice. This enables the bacteria to take up the recombinant DNA. This is not the only way to introduce alien DNA into host cells. In a method known as microinjection, recombinant DNA is directly injected into the nucleus of an animal cell. In another method suitable for plant cells are bombarded with high velocity microparticles of gold or tungsten coated with DNA in a method known as biolistic or gene gun. And the last method uses disarm pathogen vector, which when allowed to infect these cells transfer the recombinant DNA into the host. Now that we have learned about the tools for constructing recombinant DNA, let us discuss the processes facilitating recombinant DNA technology. 11.3 Processes of recombinant DNA technology. Recombinant DNA technology involves several steps in specific sequence, which as isolation of DNA, fragmentation of DNA by restriction endonucleases, isolation of desired DNA fragment, ligation of the DNA fragment into a vector, transferring of recombinant DNA into the host, cutting the host in a medium at large scale, and extraction of the desirable product. Let us examine each of these steps in some detail. 11.3.1 Isolation of genetic material DNA. Recall that nucleic acid is the genetic material of all organisms without exception. In majority of organisms, this is deoxyribonucleic acid or DNA. In order to cut the DNA with restriction enzyme, it needs to be in pure form, free from other macromolecules. Since the DNA is enclosed within the membrane, we have to break this cell open to release DNA along with other macromolecules such as DNA, protein, polysaccharide, and also lipids. This can be achieved by treating the bacterial cells or plant or animal tissue with enzymes such as lysozymes, bacteria, cellulase, plant cell, chitinase, fungus. You know that genes are located on long molecules of DNA intertwined with proteins such as histones. The RNA can be removed by treating with ribonuclease, whereas protein can be removed by treatment with protease. Other molecules can be removed by appropriate treatment, and purified DNA ultimately precipitates out after the addition of chilled ethanol. This can be seen as collection of fine threads in the suspension. Figure 11.5. 11.3.2 Cutting of DNA at specific location. Restriction enzyme digestion are performed by incubating purified DNA molecules with the restriction enzyme at a optimal condition. For that specific enzyme, across gel electrophoresis is employed to check the progression of a restriction enzyme digestion. DNA is a negatively charged molecule, hence it moves toward the positive electrode anode. Figure 11.3 The process is repeated with the vector DNA also. The joining of DNA involves several processes. After having cut the source DNA as well as the vector DNA with a specific restriction enzyme, the cut out gene of interest from the source DNA and the cut vector with space are mixed and ligase is added. This results in the preparation of recombinant DNA. 11.3.3 Amplification of gene of interest using PCR. PCR stands for polymerase chain reaction. In this reaction, multiple copies of the gene or DNA interest is synthesized in vitro using two sets of primers, small chemical synthesized oligonucleotide that are complementary to the regions of DNA, and the enzyme DNA polymerase. The enzyme extend the primers using the nucleotide provided in the reaction and the genomic DNA as template. In the process of replication of DNA is repeated many times. The segment of DNA can be amplified to approximately a billion times. 1 billion copies are made. Such repeated amplification is achieved by the use of thermostable DNA polymerase isolated from the bacterium Thermus aquatics, which remain active during the high temperature induced denaturation of the double-stranded DNA. The amplified fragment, if desired, can now be used to ligate with the factor for further cloning. Figure 11.6 11.3.4 Insertion of recombinant DNA into the host cell or organism. There are several methods of introducing the ligated DNA into recipient cells. The recipient cells, after making them competent to receive, take up DNA present in its surroundings. So if a recombinant DNA bearing gene, for instance an antibiotics example ampicillin, is transferred into E. coli cells, the host cell become transformed into ampicillin-resistant cells. If we spread the transformed cells on agar plates containing ampicillin, only transformant will grow, untransformant resistant cells will die. Since due to ampicillin resistant gene, one is able to select a transformed cell to the presence of ampicillin. The ampicillin resistant gene in the case is called a selectable marker. 11.3.5 Obtaining the foreign gene product. When you insert a piece of alien DNA into a cloning vector and transfer it into a bacterial, plant, or animal cell, the alien DNA gets multiplied. In almost all recombinant technology, the ultimate aim is to produce a desirable protein. Hence, there is a need for the recombinant DNA to be expressed. The foreign gene gets expressed under appropriate conditions. The expression of foreign gene in host cell involves understanding many technical details. After having cloned the gene of interest and having optimized the condition to induce the expression of the target protein, one has to consider producing it on a large scale. Can you think of any reason why there is a need for large scale production? If any protein encoding gene is expressed in a heterologous host, it is called as recombinant protein. The cells harboring clone gene of interest may be grown on a small scale in the laboratory. The cultures may use for extracting the desired protein and then purifying it by using different separation techniques. The cells can also be multiplied in a continuous culture system, whereas the used medium is drained out from one side while fresh medium is added from the other to maintain the cells in their physiologically most active log. Exponential phase. This type of culturing method produces a large biomass leading to a higher yield of desired protein. Small volume culture cannot yield appreciable quantities of product. To produce in large quantities, the development of bioreactor where large volumes 100 to 1000 liters of culture can be processed was required. Thus, bioreactor can be thought of as a vessel in which raw materials are biologically converted into specific product. Individual enzyme etc. using microbial plant, animal, or human cell, a bioreactor provides the optimal condition for achieving the desired product by providing optimum growth condition, temperature, pH, substrate, salt, vitamin, or oxygen. The most commonly used bioreactors are of stirring type, which are shown in the figure 11.7. A stirred tank reactor is usually cylindrical or within a curved base to facilitate the mixing of the reactor content. The stirrer facilitate even mixing and oxygen availability throughout the bioreactor. Alternatively, air can be bubbled through the reactor. If you look at the figure closely, you will see that the bioreactor has an agitator system, an oxygen delivering system, and a form control system, a temperature control system, pH control system, and sampling port so that small volumes of the culture can be withdrawn periodically. 11.3.6 Downstream processing. After completion of the biosynthetic stage, the product has to be subjected through a series of processes before it is ready for marketing as a finishing product. The process include separation and purification, which are collectively referred to as downstream. The product has to be formulated with the suitable preservatives. Such formulation has to undergo through clinical trials. As in case of drugs, strict quality control testing for each product is also required. The downstream processing and quality control testing vary from product to product. Summary. Biotechnology deals with the large scale production and marketing of products and process using live organisms, cells, or enzymes. Modern biotechnology using genetically modified organisms was made possible only when men learned to alter the chemistry of DNA and construct the recombinant DNA. This key process is called recombinant DNA technology or genetic engineering. This process involves the use of restriction endonuclease DNA ligase, appropriate plasmid or viral vector to isolate and ferry the foreign DNA into the host organism, expression of the foreign gene, purification of the gene product, the functional protein, and finally making the suitable formulation for marketing. Large scale production involves use of bioreactors.